Welcome to Physiology for Students. In this lecture, we will talk about membrane potential, specifically how we calculate it and how it changes during the action potential. During this lecture, we will define membrane potential and how it's calculated. We will describe the role of ions in the generation of membrane potential. We will then graph and talk about the phases of the action potential. We will look at how the action potential changes and common terminology related to action potentials and electrical signaling. Finally, we'll talk about action potential propagation and the role of myelin in electrical signaling. Most cells in the body use electrical signaling in some form. Classically, neurons and muscle cells of all types use electrical signaling to communicate local and long-distance signals. But there are many other cell types in the body that use electrical potentials to secrete, to create changes in the cell, to cause endocrine changes, many other things that these electrical differences across the membrane can do for cellular functions. When we measure electrical potentials, we're measuring them in electrical units of voltage. Because we're looking at very tiny cells within the body, we'll be looking at 1/1000th of a volt, or millivolt units. The way that we do these experiments is that we have a voltmeter, or a meter that's going to measure the changes in voltage, and we have at least 2 electrodes. Electrodes are the recording devices that can sense the voltage at a given point. We'll have a microelectrode just inside the cell membrane, or sometimes even kissing or touching the cell membrane. And we'll have an electrode that is a reference in the solution just outside of the cell membrane. So whenever we talk about electrical potentials, we're always comparing the inside versus the outside of the cell membrane. And that's very important to remember in terms of sign conventions. We set the outside of the membrane, or the extracellular fluid, to 0. And then all of the changes are in reference to the inside of the cell. So some membrane potential values to know and keep track of. We'll look at the resting membrane potential, or what the value of this difference in voltage is when the cell is at rest, that is, not receiving any stimulus or input. For neurons, typical value of resting membrane potential is about -80 to -70, although this can be really variable across different cell populations. If the membrane potential becomes more positive, we call that depolarizing. Often you'll see during action potentials, for example, that the membrane potential values can get more positive above 0, +10, +40, etc. So think of those depolarizing values as moving from negative to more positive. You will also hear the term hyperpolarized, and that is the value of membrane potential below rest, or more negative than the resting potential. Some negative hyperpolarization values that you'll hear may be as low as -90, -100 millivolts. We keep these ranges approximately handy so that you have these ranges in mind as we're thinking about how a cell can change during different signaling events. It's important to remember that electrical signaling in the body is driven by ions, sodium, potassium, chloride, and calcium being the most prevalent in the cells, especially sodium and potassium. We're going to focus on sodium and potassium today. Not because chloride and calcium can't change the membrane potential or they're not important for signaling, but because the sodium and potassium will give us the foundation to understand the major changes in membrane potential that happen during electrical signaling. Remember that ions cannot cross the membrane without channels or transporters. When we're thinking about ions flowing through channels or flowing across the cell membrane, we want to think about how the flow of ions contributes to current. One of the most important equations to understand changes in current and relate those to membrane potential or voltage is Ohm's law. So Ohm's law is general for electrical signaling in many different systems, and we can use it to help us understand the electrical signaling in cells. So Ohm's law, if you've memorized it in other courses, is usually written as delta V is equal to IR. And we can rearrange that to think about current specifically. So I is the current, and we can rearrange that to I equals 1 over R, or 1 over the resistance, times the voltage. So here, when we use it to understand membrane potential, I is the current or the flow of electrical charge, and that's going to be specifically the flow of charge across the membrane. V, or delta V, is going to be the difference across the membrane. This is our inside versus outside, or membrane potential difference. And then finally, R is the resistance to current flow. There's a couple of contributors to resistance in a cell. The internal resistance of the cell is essentially the size of the cell. Smaller cells will have higher resistance, and larger cells lower resistance to ion flow. We generally don't think of those very much or change those. Really what we want to think about for resistance in the cell membrane or at the plane of the membrane is going to be whether the channels are open and how open those channels are. So the resistance is basically the inverse of the permeability or conductance, whether the ions can flow across the membrane or whether the channels are open for any particular ion. It's really important to think about ion permeability. How easily can this particular ion or this group of ions cross the membrane? If the channel's closed, they can't cross at all at that given point in time. If the channel is partially open, they have a small permeability. If the channel is fully open, we'll have a fast conductance or a large permeability. So I've used the term permeability and conductance. So permeability is how easily an ion can cross the membrane, and that's measured in electrical conductance. Electrical conductance is essentially how much can flow through a channel at any given time. So it's given in coulombs per second per volt, or per voltage change in the membrane. So that is how much can cross given a particular voltage change. So remember and picture the changes as these channels for the ions are opening and closing. When the channels are open, you can have a large conductance. When the channels are closed, you have a conductance that is approaching 0. So let's think about the channels and pumps that are going to be important for driving the membrane potential. First, we have to give credit to the sodium-potassium ATPase. The sodium-potassium ATPase, or the sodium-potassium pump, is what sets up the sodium and potassium gradients across the extracellular and intracellular compartments. So remember that the sodium-potassium ATPase is a primary active transporter, meaning that it directly hydrolyzes or uses ATP to pump sodium and potassium against their gradients. And it always pumps 3 sodium out into the extracellular compartment and 2 potassium in to the intracellular compartment. So it swaps sodium and potassium and uses ATP to do that. This is what sets up the initial differences in sodium and potassium inside versus outside of the cell. The net effect is that there's more positive charge in the extracellular fluid portion of the membrane than the intracellular portion of the membrane. And that is that positive charges are building up outside the membrane and negative charges are building up inside the membrane. And remember, when we have a difference in charge, that's going to generate a membrane voltage. So let's focus on potassium and sodium and look at some values. This is all in Chapter 5 of the Guyton textbook, and I'm using the concentrations given in Chapter 5 so you can practice the calculations. Depending on the textbook, depending on the cell type you're looking at, the concentrations may be different. So you don't need to memorize the concentration, but generally look at the process of how we're calculating and finding these membrane potentials. So, potassium. Potassium is always higher inside the cell compared to outside the cell. Why? Because of the sodium-potassium pump. The values given in the Guyton textbook are about 140 milliequivalents per liter inside the cell and 4 milliequivalents per liter outside the cell. Remember those. We're going to use those in our calculations in just a few minutes. We also want to ask, for permeability purposes, what channels should we think about when we're thinking about potassium in neurons, particularly related to action potentials? There's a number of channels for each of these ions, but today we're going to focus on the channels involved in the typical action potential. So the channels related to the action potential are, there are potassium leak channels. These channels are always open and they're permeable at rest, primarily to potassium, but also to a little bit of sodium. They're about 100 times more permeable to potassium than to sodium. That's why we call them the potassium leak channels. We also have voltage-gated potassium channels. Voltage-gated potassium channels are only going to open when the membrane potential increases or depolarizes from rest. So the voltage-gated channels are going to be closed until the cell receives an input. When the cell receives an excitatory stimulus, these voltage-gated potassium channels will then open and lead to changes in potassium permeability. One important property of the voltage-gated potassium channels is that yes, they open when the cell is stimulated, but they're very slow, much slower than the sodium channels. So it takes a little while before the potassium conductance kicks in when the cell is stimulated. Remember that, that's going to be important when we graph our action potential. So these channels are slow. Now let's look at sodium. Again, the sodium gradient is established by the sodium-potassium pump. The values given in Chapter 5 of Guyton are 14 milliequivalents per liter inside the cell and 142 milliequivalents per liter outside the cell. In other words, there's a lot more sodium outside the cell than inside the cell. The channels related to action potentials for sodium are voltage-gated sodium channels. These channels open when the membrane is depolarized up to approximately a level of minus 55 millivolts. So remember, we gave a typical membrane potential at rest of minus 80 to minus 70, and as that moves more positive with different stimuli coming in, those sodium channels will open around minus 55 millivolts. That's very important. They open quickly, and as they open quickly, they're going to allow sodium to enter into the cell and depolarize the membrane. So these channels are fast, much faster than potassium channels, and they open due to changes in membrane potential. They also have two gates. They have an active activation gate that opens at this minus 55. They also have an inactivation gate that is time dependent. In other words, it closes shortly after the activation gate opens. So often we draw the sodium channel with two gates. Here's the activation gate, here's the inactivation gate. Think of this inactivation gate sort of like a plug that will plug up the channel after it's been open for a very short period of time. So the activation gate opens, and then shortly thereafter, the inactivation gate closes the channel. It's also important to note that the inactivation gate cannot be removed or reset until the membrane voltage repolarizes back down to its resting state. So it must be in a repolarized or low membrane voltage, highly negative membrane voltage, for this to reset. It resets at negative membrane voltage. To really understand how sodium and potassium or any ion contributes to the membrane potential at a given period of time, you will want to calculate the Nernst potential for each ion. Here we use the Nernst equation. So the Nernst equation finds the voltage of the membrane at which the electrical and chemical forces are balanced. It's not simply the concentration gradient that tells us whether an ion will move in or out of the cell. We need to look at both the chemical and the electrical forces and calculate the Nernst potential. So what we do for the Nernst potential is we imagine, what if the membrane is fully permeable to only one species of ion? Let's say only potassium or only sodium at a given time. What would the membrane potential be if that ion were allowed to reach equilibrium? We then calculate that value, and that gives us the equilibrium potential, say for potassium, which is the Nernst potential. I have a separate video in which I calculate the Nernst potential, and you can watch that if you'd like some practice with calculating the Nernst equation. We simplify the Nernst equation to the equilibrium potential for an ion is negative 61 over Z, which is the charge of the ion, times the log base 10 of the concentration inside the cell versus the concentration outside the cell. For sodium and potassium, Z is +1, so that makes it very simple. And then you just have to plug in the values, millimolar values, for inside versus outside the cell. So pause here and see if you can calculate the Nernst potential, or the equilibrium potential, for potassium and the equilibrium potential for sodium, given those concentrations from Guyton chapter 5. So can you calculate EK? And can you calculate ENA given that Nernst equation? Pause, and I'll give you an answer in just a second. Okay, so were you able to do it? Let's take a look. The E ion is going to be equal to negative 61 over Z times the log of the inside versus the outside concentration. So for EK, that corresponds to negative 61 over 1 times the log of 140 over 4. And that gives us about negative 94. Is that the number you got? About negative 94 millivolts for EK. Okay, let's try ENA. For ENA, we get negative 61 over 1 times the log of 14 over 142. Those are the given values in the textbook. That gives us, when you calculate that through, an ENA, or an equilibrium potential for sodium, at positive 61 millivolts. So the equilibrium potential for potassium is very negative, and the equilibrium potential for sodium is highly positive. These ions have very different effects on membrane potential when their channels are open. So the way we think about this is, at a membrane voltage, our typical membrane resting potential of minus 80, with only potassium channels are open, they will hyperpolarize the membrane, bringing it from minus 80 down towards minus 94 millivolts. For sodium, at a typical membrane potential of minus 80, if only sodium channels are open or the sodium channels are dominant, they're going to depolarize the membrane up towards positive 61 millivolts. So let's look at some values at different times of the membrane. So I've told you that the resting membrane potential is usually minus 80. So now that we know the equilibrium potentials for potassium and sodium, which ion do you think is dominant at rest based on the information we've given you? If you said potassium, you're correct. Did you notice that at rest, the membrane potential is very negative, minus 80 millivolts? The potassium leak channels are open at rest and much more permeable than sodium channels. So that means that potassium is driving the membrane potential towards its own equilibrium, towards that minus 90, 94 millivolts. But it doesn't quite get there because there are other ions that have slight permeabilities in the membrane at rest. So the equilibrium potential for potassium is dominating during the resting phase, meaning when the cell is just sitting there waiting for a stimulus. So there are other ions that are contributing, which is why it's not identically the equilibrium potential for potassium, but it's very close. So if we want to know the resting potential for any given cell, we will ask first, what are the ion gradients at rest? And second, how permeable is the membrane to that particular ion at rest? And that is, which channels are present and are they open at rest? Ultimately, this will give us our calculation of resting membrane potential. If you want to get really fancy and say, I'm not satisfied with this, minus 80 is sort of close to EK, what's really going on with all of the other ions? If you really want to know how the other ions are contributing to the full resting membrane potential, you would use something called the Goldman Equation, or the Goldman-Hodgkin-Katz equation. This is an equation that accounts for each ion's concentration, and each ion's permeability to give the full resting membrane potential. So let's take a look at this equation. This one, again, you'll see that negative 61 over Z times the log, but this time you're going to see values of concentration and permeability for each of the ions. So here we can see permeability and concentration gradients for sodium, permeability and concentration gradients for potassium, and permeability and concentration gradients for chloride. We could continue this equation if we knew that other ions were involved. For example, if we knew that calcium was involved in a particular cell, we could plug those into the equation as well. Now, if you plug in all of those numbers and the permeability constants for the membrane for those known channels, then you'll get the exact membrane potential at rest. So we need to know the permeability and the concentration inside versus outside for each ion to calculate the Goldman equation. I don't force my students to calculate the Goldman equation, but I do think that it's really helpful to at least see it so you can say, okay, this is how I would know how a particular ion species was contributing to the membrane potential. Okay. Let's talk about some terminology. We've said some of these terms already, but let's just make sure we're all on the same page. So here, if we're graphing membrane potential in millivolts, if we look at 0, so we set a point of 0, that would be a non-polarized membrane. There's no difference between the inside and the outside of the membrane. If we're anywhere that's non-zero, then that would mean that the membrane, in fact, is polarized, which is the case for body cells. All membranes are polarized to some extent. Neurons, muscles, and excitable cells are highly polarized or far from zero at rest. So as we said, a typical resting membrane potential for a neuron is around -80. So that's very polarized in the negative direction away from 0. So if our resting membrane potential is -80, now we can compare all other time points to that resting membrane potential. If a membrane potential during a stimulus moves more positive from the resting membrane potential, meaning on this graph that it would move up, we would say it's depolarized, or moving towards 0 and even past 0, more positive. If we move lower than the resting membrane potential or more negative, we would say it's extrapolarized, or moving further from 0, hyperpolarized. So if we move in the downward direction from the resting membrane potential, that's going to be hyperpolarized, or even further from 0. So keep these in mind. We're going to use the terms depolarized and hyperpolarized a lot as we discuss the action potential. Okay. Before we get to action potentials, we need to see what leads up to an action potential. What leads up to an action potential are graded potentials. Graded potentials are small stimuli, small inputs to a cell, to a neuron specifically. So graded potentials are very small changes in the voltage of the membrane that occur when stimuli open ion channels. In terms of a neuron, these usually occur at the dendrite or at the cell body of the neuron. So here we have a neuron, typical pyramidal neuron where we've got dendrites at the input side. And here's our cell body. And then the axon and the axon terminals. So typically our graded potentials are going to be happening at the dendrites and the cell body. They're small, they're local, and they dissipate over time, meaning that they don't last very long. When I say small, you can think of graded potentials as being somewhere between 1 and 5 millivolts. Although again, that can vary based on the cell and based on the input. So these are small stimuli at the dendrites and cell bodies of these excitable cells. It's also important to note that graded potentials can be either excitatory, depolarizing, or inhibitory, hyperpolarizing. Depends on the neurotransmitter and it depends on the receptor present on the dendrites or the cell body. So it depends on the input, and it depends on how that input is received at the dendrite or the cell body. For example, if there's a particular neurotransmitter receptor combination on the dendrite that happens to open sodium channels, what would happen? Well, those sodium channels will open and depolarize the membrane of that dendrite in that small local space. What would happen if instead the particular combination opens potassium channels? Well, as we've just learned based on the Nernst potential, potassium permeability causes a hyperpolarization of the membrane. That would be an inhibitory or a more negative stimulus to the cell. So for any given cell, if we're looking at a graded potential, what we want to ask is, how does it respond to this stimulus? Does it cause the channels to open? Does it cause the channels to close? Which channels will open in response to the stimulus? Does it open channels that would cause depolarization, or does it open channels that would cause hyperpolarization? If the channels cause depolarization, they're called EPSPs, or excitatory potentials. If the channels cause hyperpolarization, they're called IPSP, or inhibitory potentials. Why do we care about graded potentials? We care about graded potentials because they are what will add up to these larger action potentials. So we have what's called summation or adding of graded potentials. So graded potentials can occur together in time, which would be a temporal summation, or they can occur together in space on the dendrites, which would be a spatial summation. If you get two graded potentials that are excitatory in time or in space, then they're going to add up to a bigger stimulus to the neuron. If you get two graded potentials that cancel each other out, then effectively you will have no stimulus to the neuron at that particular point in space or time. So it is the graded potentials that are going to add up to lead to either cancellation of the stimuli or increased stimuli that it can eventually lead to action potentials. So if they add up to depolarize the membrane to a level in which we call threshold, then the positive feedback cascade of an action potential will be triggered. So let's talk about threshold. Threshold happens when the voltage-gated sodium channels are triggered to open. For most neurons, that's at a value of minus 55 millivolts. So let's take a look. So if we're looking at our membrane potential and the neuron starts at rest at minus 80, you would have a certain number of graded potentials adding up to get to a value of minus 55. If you get enough graded potentials to add up to minus 55, then you've hit threshold, and that triggers the voltage-gated sodium channels. Once the voltage-gated sodium channels are triggered, we're going to have an all or nothing pattern of action potential that follows. If you don't get to threshold, you don't have an action potential, and you simply go back to the point where you're adding up graded potentials until you can reach threshold. So once it triggers, then we'll get a full completion of the action potential. So threshold is an incredibly important concept with respect to action potential. So what happens at threshold? The opening of voltage-gated sodium channels. Two key terms related to this. It's an all or nothing pattern. If you hit threshold, you get an action potential. If you don't, you get nothing. And we'll also talk about propagation, which is the spreading of the action potential down the axon of the neuron. We're going to take a minute to graph the action potential. If it's been a while since you've seen the action potential, we'll review the pieces here. Get a piece of paper and probably a couple of colored pens, and we'll graph the action potential together. Okay, so here's the action potential. Let's start by setting our axes. So on the y-axis, we're going to have millivolts, which is our change in membrane potential. And on the x-axis, we're going to have milliseconds, our change in time. Let's set some boundaries for our action potential. What boundaries do you think you should set for the action potential based on what we've talked about so far? If you said resting membrane potential, yes. If you also thought equilibrium potentials for potassium and sodium, extra yes. Good work. So we can't go any lower than the equilibrium potentials for our key ions or any higher. So the lowest we could go would be EK, which is at minus 90. The highest we could go would be ENA, which is at positive 60. But you know that we're going to actually start at our resting membrane potential. Our resting membrane potential is about minus 80 for most neurons. So the cell is sitting at rest. It's waiting for a stimulus. It's reached its value of resting membrane potential based on the ion gradients and the permeability of the channels at rest. Then it's going to get some graded potentials. Some might be excitatory, some might be inhibitory. If we get enough to add up, we're going to reach threshold. I'm going to remove those graded potentials so you don't think they're part of the action potential. So let's back up a little bit. Okay, so let's say we've had depolarizing graded potentials that are going to depolarize the membrane to what level that's important? To threshold. Threshold is minus 55. So let's draw a dotted line for threshold. If we get enough graded potentials to add up to threshold, the voltage-gated sodium channels will open, and the membrane potential is going to be driven towards the equilibrium potential for sodium. Those channels are very fast, and they're depolarizing. So this is going to be sodium entering the cell and depolarizing the membrane. But there's a limit. We don't reach the full equilibrium potential for sodium. And it's limited because of that second gate on the voltage-gated sodium channels, the inactivation gate. So the peak of depolarization of the action potential is limited by inactivation of the sodium channels. That inactivation gate closes the sodium channels, and the rising phase of depolarization stops. Now remember that the potassium channels were slow to open, so they're a little bit late to the party, but they're open by the time the inactivation gate closes for sodium. So now the potassium channels will be the drivers of membrane potential, and that's going to be potassium leaving the cell. When potassium leaves the cell membrane, that's going to repolarize and eventually hyperpolarize the membrane potential. Why? Because the equilibrium potential for potassium is negative based on its electrochemical gradient. So potassium then drives the membrane potential more negative back towards rest. And now we have the falling phase of the action potential. They stay open a little bit longer because they're also slow to close. And we get what we call the after hyperpolarization of the action potential, and that is it hyperpolarizes beyond rest as the voltage-gated potassium channels stay open a little bit longer. The membrane potential then returns back towards rest as the voltage-gated potassium channels close. So the voltage-gated potassium channels close. You still have the leaky potassium channels, which establish our membrane potential to begin with. It's also important to note that at this time, as the membrane potential is going back down towards rest, that the sodium channels are able to reset. That is, the inactivation gate that is plugging the sodium channels resets and allows the sodium channels to be ready for a new stimulus to come in. Now the membrane is back at rest and ready for a new action potential to be generated given enough stimuli. Okay, so review the values here that we put on our action potential graph. Can you graph these yourself and explain the rising phase, the limited peak, the falling phase, and the after hyperpolarization phase of the action potential based on the behavior of the channels for sodium and potassium? Pause, make sure you can do that, and then we'll move on to a couple of specifics. Okay, some specifics about the action potential. First of all, we have something called a refractory period. There's two refractory periods to note for the action potential. The first is an absolute refractory period. This is a point in time at which all the sodium channels are either fully active, meaning no more can be activated, or they have been inactivated at that peak of the action potential. At this point in time, no additional stimuli can excite the neuron to cause a second action potential. So the absolute refractory period means absolutely no further action potentials could be generated at this time. The relative refractory period is a little bit different. It's a little bit later in the action potential. This is the point at which the sodium channels are reset, but the membrane potential is farther from threshold than it normally is at rest. This is basically, you could get an action potential, but it's more difficult relative to if the neuron was at rest. So take a look at your action potential drawing. Can you find these time points on your drawing? Let's take a look at mine, see if we can find them there. Let's make a little space here. So the absolute refractory period would be about this point in time. Before the sodium channels have reset. So in other words, the sodium channels are either maximally active or they've been inactivated by the inactivation gate. So I picture the channels, sodium channels like this, with their activation gates open and the plug of the inactivation gate closing those channels. The relative refractory period would happen at about this point in time. When the sodium channels have reset, but the after hyperpolarization is occurring and the membrane is further from threshold. So here we can picture the sodium channels as having their activation gate closed and their inactivation gate open. But remember, the membrane is also hyperpolarized at this point, leading to it being more difficult to get to threshold to generate that next action potential. So the relative refractory periods are important for limiting how many action potentials can occur in a given time frame. Later on in the unit, we're going to talk about other action potentials that look different from the action potential that we've drawn today. For example, some action potentials don't have this sharp peak that we just drew, but instead they have a long, flat plateau or a prolonged depolarization phase. What's happening during this prolonged depolarization phase? Well, it must be an ion, a positive ion, that is entering the cell. So in this case, these are present in the cardiomyocytes of the heart, and they exhibit this long plateau because of L-type calcium channels. These channels are slow to open, voltage-gated calcium channels that allow calcium and sodium to enter the cell membrane at this point in time. And they cause an extended phase of depolarization and a very unique action potential shape for those cells. Some cells also have spontaneous action potentials, meaning that they don't actually sit at rest. That is, these cells, particularly cardiac pacemaker cells, peristaltic cells in the intestine, they actually have a continuous generation of action potentials. So they never really get fully down to rest. Once they get down to a low resting membrane potential, close to about -60 to -80, they actually trigger a different set of channels. In the heart, those are called funny sodium channels that open at rest to depolarize the membrane to threshold. So instead of waiting for an outside stimulus, they actually have their own method of depolarizing the membrane at rest, constantly activating rhythmic action potentials. This is important for the rhythmicity of the heart and also for the rhythmicity of intestinal peristaltic waves, for example. So these are some unique types of action potentials. All right, finally, we're going to finish up by talking about propagation of action potentials. So one of the most important parts of an action potential is that it travels down the axon to excite the axon terminal, or the end of the neuron, leading to communication between one neuron and the next. So it's important that action potentials spread down the membrane to transmit these long-range signals. How do they do that? They do that because the sodium activation depolarizes and excites nearby channels down the axon. Because of the inactivation that happens very quickly, once sodium channels are activated, they quickly inactivate, but then they've excited the ones next door. And that continues in a positive feedback, sort of a domino effect, down the membrane. That causes the propagation of the action potential down the axon, and it occurs in a one-way direction. They can't go backwards because of the inactivation of the sodium channels behind them. So this is a positive feedback loop that continues propagating the action potential down to the axon terminal. So remember, when we're looking at a neuron, that the signaling is directional. The graded potentials come into the dendrite or to the cell body. They add up in a region that is rich in sodium channels called the axon hillock. If you get enough stimulus there to trigger the voltage-gated sodium channels in that area, then you'll continue to trigger voltage-gated sodium channels all down the axon until you reach the end of the axon where a neurotransmitter will be released, causing communication signal to the next cell. Another thing that helps propagation is myelination of certain axons. Not all axons are myelinated. Usually the axons that are myelinated are the axons that are traveling long distance. For example, motor neuron axons that are traveling long distance down the body. So the axons of some neurons can be wrapped in myelin. If they're in the periphery or the peripheral nervous system, Schwann cells are responsible for wrapping those axons in myelin. If they're in the central nervous system, oligodendrocytes are responsible for wrapping those axons in myelin. Myelin is a thick, insulating layer containing sphingomyelin. That sphingomyelin insulates portions of the axon that's drawn here in this grayish blue color. So here we're looking at a Schwann cell wrapping around the axon of a peripheral motor neuron. They wrap around and create this insulation. You might be thinking insulation, that's supposed to prevent charge from moving, and that's true. So what they do is they actually have areas of myelin and areas in between where there's no myelin. The areas where there's no myelin are called the nodes, or the nodes of Ranvier. These are areas that are highly concentrated with sodium channels in between areas of myelin. How does this work? The way that it works is that the myelin insulates and the nodes of Ranvier conduct very quickly the signals. So think about if you're walking across a garden. You can either saunter step by step across the grass, or you can hop along stepping stones. So the way that the action potential propagates very quickly down these myelinated axons is by hopping quickly from node to node, skipping regions and moving very fast down the axon. That conduction very fast from node to node is called saltatory conduction, and it's 5 to 50 times faster in myelinated cells than unmyelinated cells. So myelinated neurons are neurons that need to signal fast and across long distances. They use this saltatory conduction to very quickly conduct their action potentials from node to node, skipping from one end of the axon to the other end of the axon to propagate that signal all the way down to the next cell. All right, that's it for membrane potential and action potentials. I hope this was helpful. Please let me know if you have any questions.